welcome back class 9 to the online classes. We had already started the first chapter of the cell and we are talking about the various cell organelles. What is cell organelles? Once again I will remind you, the living parts of the cell, just like our body has many organs that work together to perform a particular function. Similarly, inside a cell, very tiny organs are there which carry out different types of functions, like endoplasmic reticulum we studied last. So today we will start with the Golgi apparatus. Okay. Okay, now listen this. Golgi apparatus, it is also present in the eukaryotic cells. It is found in eukaryotic cells. Okay. It is also membranous lamellar structure. I am not writing all this. It is membranous lamellar structure which is found in the eukaryotic cells. It was discovered by Camillo Golgi. Whatever is important, that only I will write because we have plenty. Camillo Golgi. It was discovered by Camillo Golgi in 1838, and that's why the name is Golgi apparatus. It is also known as Golgi bodies, Lysosomes, Golgi structure, Golgi membrane. So many names it is called. Okay. Now next point you can write that Golgi apparatus in plant cells, it is known by a special name called dictyosomes. So what is dictyosomes? It is name given to the Golgi apparatus which is found in plant cells. It has a special name in plant cells. Okay, dictyosomes in plant cell. In plants it is found. Okay, Golgi apparatus is known by this name, Golgi dictyosomes. Now next one is Golgi apparatus. From where does it originate? It originates from the endoplasmic reticulum only. The budding of endoplasmic reticulum, that is the vesicles which are present, the last part, they give rise to the Golgi apparatus or the Golgi bodies. Now what is the structure of Golgi bodies? If you see the structure, you will find that it has parallelly arranged stacks. Stacks means like this. This cisternae is there. Like it is made up of structurally three parts, like we had studied in endoplasmic reticulum. What are they? Cisternae, tubules, and the third one is vesicles. They are the three structural components of Golgi apparatus. So like this parallelly arranged stacks of membranous lamellar structure is there. Okay, it is unbranched. Like this I have drawn. Okay, now from here I will draw the vesicles are there. A very simple structure of Golgi apparatus. Now inside it, whatever gap is there, that is called the lumen, L-U-M-E-N. And this face, the upper face, it is called cis face. The lower face, this is known as trans face. So what are these? These two are the faces, cis face and trans face. This is the newly forming phase, and this one is the maturing phase, the matured phase. Okay. So lumen is the gap which is present inside the lamella. So what are they called? They are known as lamella. Okay, stacks of lamella. So stack means like one arranged on top of the other. This is called stack of lamella, and they are unbranched. They are membranous also. They have a membrane also. No. Now, so this is the structure of. So what is cisternae? Already I said. What is tubules? Tubules are branched network which arises from it. Like from here, if the network arises, okay, that will be branched. That is called tubules. The tiny tubes are known as tubules. And vesicles, like these are the structures which is called vesicles. Okay, the one which is budded up from the cisternae only. Like this part is cisternae. From here it is cut. Then they will form the vesicles. Okay, and these vesicles contain plenty of enzymes inside it, which are hydrolytic enzymes also, which has different types of functions to do. Enzymes, hormones, many things are present in it. So cis and trans phase are the two phases. Cisternae, tubules and vesicles are the three parts or the structural components of Golgi bodies. Okay. And now next, what is vesicles? Already I said that vesicles are membranous sac-like structure that contain different types of enzymes, secretory products, and they are originated from the cisternae and tubules only. Okay. Now next we have to study that what is endomembrane system within the cell. The Golgi apparatus, along with or in association with endoplasmic reticulum, lysosomes and the other cell organelles, it forms a network. That network is called endomembrane system. endomembrane system. Okay. So if it is asked what is endomembrane system, endomembrane system is a network of organelles which are present inside a cell that includes endoplasmic reticulum, Golgi apparatus, lysosomes, nuclear membrane, cell membrane, etc. Okay, now next, what are the functions of Golgi apparatus? The functions of Golgi apparatus, you remember that in the plant cell, at the time of cell division, a cell plate is formed, like if this is the plant cell, during cytokinesis, when the cytoplasm of the plant cell divides, this cell plate is formed due to Golgi bodies only. So formation of cell plate in plant cell during cytokinesis. This is one of the function. Second function, the acrosome at the tip of the sperm cell is there. This tip contains acrosome. Okay. So what is this acrosome? Acrosome, or which is found in the head of the sperm cell. It also contains, or it is made from the Golgi apparatus, which contains enzymes to dissolve the wall at the time of fertilization. Now next one is, what is the other functions of Golgi apparatus? It is the one which helps in condensation, packaging. So what I said, it helps in condensation, packaging and transportation. Three words I said. It helps in condensation, packaging and transportation of different types of biomaterials. So different types of biomaterials can be transported through this Golgi apparatus. And Golgi apparatus is the one which helps in, like, it contains different types of enzymes, alkaloids, hormones and many more chemicals which are dissolved inside, which are present inside the vesicles. Apart from that, it helps in the synthesis of polysaccharides. What is polysaccharide? It is a type of carbohydrate, pectins and other carbohydrate. Not only carbohydrate, it helps in the synthesis of phospholipids as well as glycolipids. That is, glycolipids and glycoproteins are also synthesized. It helps to form the acrosome in the head of the sperm cells. So this much only about the Golgi apparatus. Okay. Now next cell organelle that we are going to study is ribosome. Okay. You have heard about the ribosome. Like, it is commonly called as protein factory of the cell. Why? Because it helps in the synthesis of proteins inside a cell. That's why ribosome is called protein factory of the cell. Now, where are these ribosomes present? Ribosomes are globular structure, and whether they contain membrane or not, they are membraneless. Membraneless means, if this is a ribosome, they do not have any covering around it. This one is not there. So next we are studying is ribosomes, the next cell organelle. Ribosomes are membraneless. What are some of the characteristics? Membraneless means without membrane. You can also write without membrane. Smallest means very small, or the tiniest organelle, cell organelle. Okay. Membraneless, smallest cell organelle, which is present, you may have seen, it is found on the outer surface of endoplasmic reticulum. These are the ribosomes. This is endoplasmic reticulum, and such endoplasmic reticulum that contains ribosomes on its outer surface, they are called rough endoplasmic reticulum. So these ribosomes are present in on the outer surface of endoplasmic reticulum, and it is made up of rRNA as well as proteins. So what are the components of RNA? It is made up of rRNA and R proteins. R means, here small r I am writing, rRNA, that is ribosomal RNA, and R proteins, that is ribosomal proteins. They are the one which makes ribosomes. Ribosomes are made up of rRNA and R proteins. And this ribosomes is made up of two units, smaller units. Like if I draw like this one is bigger one, then this one is smaller one. Two sub-units are there. What are they? They are made up of two sub-units. Sub means smaller parts. Like 60S, 40S. So two sub-units are there. And you know that two different types of cells are there. Prokaryotic and eukaryotic. Prokaryotic are the primitive cells, the one which is found in bacteria, blue-green algae. That type of cell is called prokaryotic. So it is made up of two subunits and they are called the structural subunits. Prokaryotic ribosome is 70S type. So if it is asked what type of ribosomes is found in prokaryotes, it is 70S. I am writing this capital S. Now what does this S indicate? The S stands for Bedworks unit. SBEDBERGS, Bedworks unit, which denote the sedimentation coefficient. Okay, so what does S indicates? S indicates Bedworks unit. Just like in maths also we have units like kilometer, centimeter, they are the units kilogram. Okay, temperature is measured in degree. No, so similarly here it is the Bedworks unit which is used to indicate the ribosomes. So 70S type of ribosomes is found in prokaryotes. In eukaryotes, the type of ribosome which is found, it is called ATS. ATS type of ribosomes are present. Okay, ATS. Now this 70S type of ribosomes, it is made up of two structural components and they are 50S and 30S. Now see, you will be confused that I have written 70 here and writing that it is made up of two parts, 50 and 30S. 50 plus 30 becomes 80S. It's not like that math calculation we have to do here. Okay, 50 plus 30, 80. No. And here the ATS in eukaryotic ribosomes, it is made up of 60S plus 40S. They are the two subunits. But if you count plain like maths, we are doing 60 plus 40 becomes 100. But here it doesn't apply. Okay. So no need to count in that way that 60 plus 40, sir is writing 80 here. It's wrong. So ATS type consists of two subunits. The larger unit is 68, like this one is the larger A. So I can write here this one 60S, then this one 40S. Okay. This is larger subunit, means the bigger part of the ribosome. This is the smaller part of the ribosome. And this type of ribosome is found in eukaryotes, eukaryotic ribosomes. Okay. So what is eukaryotes? Eukaryotes, you know, all the modern plant and animal cells are called eukaryotic cells. Okay. And their diameter may be 2.5 to 2.8 nm. Nm means nanometer, the diameter. Like if I draw this one as the ribosomes, the diameter may be 2.5 from here to here to 2.8 nm, nanometer. Okay, this much very small length is there. Nano means very small size it is. So we have studied about the, now what is the function of ribosomes, that we have to do. Ribosomes, where they are produced? They are produced inside the nucleolus. It is the nucleolus. You know what is nucleolus? It is the name of the organelle which is like, this is the nucleus. The small spherical bodies which are present inside the nucleus, they are called nucleolus, and they give rise to the ribosomes. Hope you have understood this one. Now next cell organelle that we have to study is lysosomes. You have heard no lysosomes. It is also popularly known as the suicidal bags of the cell. Why it is called suicidal bags? Because it contains enzymes, and sometimes in pathological conditions, the enzymes help in the digestion of the cell organelles. It digests its own cell organelles, and that's why it is called autophagy also. Okay, so next cell organelle is lysosomes. Lysosomes was discovered by D. Duke, the name of the scientist, D. Duke, Christian D. Duke, in 1955, the year I have to mark. D. Duke in 1955 discovered lysosomes. And what are they? Do they contain the membrane or not? No. They are also spherical or ovoid, single membrane bound. So they are spherical, means round in shape, or ovoid, single membrane bound organelles, means only one membrane is there. Single membrane bound organelles. Where are these organelles present? These organelles are found in plant cell or animal cell. They are present in animal cell. They are not found in plant cell. Okay? They are found in animal cells. They are absent in plant cell. Not in plant cell also you can write. It is found in animals, but it is absent in plants. It is a cytoplasmic organelle. Now next, lysosomes. Who discovered it? This one you can learn. D. Duke. In which year? 1955. What is the structure? It is spherical or ovoid in shape. And how many membranes are there? It has a single membrane, one membrane is there, not like mitochondria or chloroplast in which double membrane is present. And where it is found in animal cells, not in plant cells. Now next, what is the chemical composition of lysosomes? Lysosomes contain 40 different types of enzymes. 40 types of enzymes are there. Now what are the names of some of these enzymes? If you ask example, it consists of the enzymes like nucleases, phosphatases, lipases, okay, then what we can write? Nucleases, phosphatases, lipases, then this one, nucleases means the enzymes which are present inside the nucleus, phosphatases, phosphate containing enzymes, and lipase, you know what is lipase? It is the name of the enzyme which helps in the digestion of fats. Synthetases also we can write, synthetases, okay. So many more are there. Proteases, nucleases, glycosidases, phosphatases, sulfatases also you can write, sulfatases. Now 40 different types of enzymes are there. Only some of the names I have written here: nucleases, phosphatases, lipases, synthetases, sulfatases, okay. All the enzymes ends in ASE, you can see ASE. That is the suffix of the enzymes. Means wherever you write the name of the enzymes, at the last there will be ASE. That denotes the suffix, means the last 3 letters of that word which ends in ASE indicates the name of a enzyme. And these enzymes are useful for digestion of both extracellular and intracellular materials. Suppose a cell is there. If any material is present here, it is the lysosomes who will digest them. And this type of digestion of the food material or any other waste materials present outside the cell is called heterophagy. Heterophagy means the one in which non-cellular materials, this is a non-cellular material, it does not belong to the cell, non-cellular material is digested by the lysosomes of the cell. So that type of digestion is called, the word is called heterophagy, P-H-A-G-Y. Hetero means different, phagy means to eat. So if a cell is eating something which do not belong to the cell, means any outside material, that is called heterophagy. Got it? Now one more is there, autolysis or autophagy. Autophagy, P-H-A-G-Y. What is the meaning of autophagy? Autolysis or autophagy. Auto means self, phagy means eat. So if sometimes in pathological condition, the enzymes which are released from the lysosomes, they digest its own cellular organelles. Means within the cell only, the materials like mitochondria, ribosomes, they are digested. So they will die. Because if they kill their own cell parts, that will result in the death of the cell, and that is called autolysis or autophagy. And that's why they are also known as suicidal sac or suicidal bags of the cell. Means they are the one which can cause suicide of the cell due to the presence of digestive or hydrolytic enzymes. Got it? Now one more function of lysosomes is there. This one you can write it as function also. The third one is, it is believed that it is lysosomes which initiate means start the process of mitosis, cell division. So they start mitosis, cell division. Start means initiate. The mitosis, cell division starts due to the presence of lysosomes. Okay. So now next we have centrosome, the next cell organ. You know what is centrosome? Centrosome is the name of the cell organ which is made up of 2 centrioles, and that is present only in the plant cells near the nucleus. It is a clear material present near the nucleus. It is visible during the interphase. So what is centrosome? Centrosome is a clear area near the interface nucleus of an animal cell. A clear area, it is indicated by, the centrosome is indicated by, or its presence is known by a clear area near the interface nucleus of an animal cell. It is found only in the animals. Centrosome is the characteristics of animal cells only. It is not found in plant cell. And it is made up of like two centroids. Two cylindrical bodies are there, like one, I will draw it here like this. They are called the microtubules. Two cylindrical structures are there. Both of them are arranged perpendicular to each other. Like if one of them is like this, the other one will be like this. Okay? At 90 degrees. So one of them, this is called centroid. The other one will be arranged in this way, in this direction. Okay? Now see, what is their position? They are perpendicular to each other, 90 degrees. So both of them are called centroids. So what is centrosome made up of? Centrosome is made up of two hollow cylindrical structures which are known as centroids. And they are made up of, each one of them, it is known as, the name of this is microtubules. Microtubules. These microtubules play a very important role, and it attach with, like this is the centromere. So microtubules will attach here at the centromere of a chromosome, and it will help in the separation of chromatids during cell division. Okay? So what is centrosome? What is the function of centrosome? What is it made up of? Where it is found? Everything I said. Who discovered it? One scientist named T. Boveri in 1888. T. Boveri in 1888. Name of the scientist who discovered centrosome. T. Boveri in 1888. What is it? It is a membraneless, there is no membrane around it, so membraneless cytoplasmic organelle which is present near the nucleus of animal cells. It is absent in plant cell. It is absent in plant cells. In plant cells it is not found, okay? This particular cell organelle. And what is it made up of? It is made up of, this also known as diplosome, the other name, also known as diplosome. Why? Because it consists of two cylindrical structures which are known as centroids. That's why the word is diplosome. Okay? Now next we have to do is plastid. Okay, now next we have to start is plastids. I already said plastids are double membrane bound. Double membrane means they have two membranes, two coverings around it. So double membrane bound, I am writing here double membrane bound means containing two membranes in simple word. Double membrane bound cell organelles. These cell organelles are present only in plant cell, and they are of different shape and size. Okay, they may be star shaped, they may be ribbon shaped, found in plant cells only. Found in plants only, I am writing. Okay, plant cells means plants only. And there are three types of plastids according to their color, what color they impart. Three types of plastids are there. What are they? I'll just write the name and the color which they give. Number one is chloroplast. The green colored plastid is called chloroplast. So I am writing the color here, green color. Number two, chromoplast. Chromo means the colorful plastic, the one which gives color to the flowers and fruits. So here we can write colored, colorful or colored. Different colors are there. Yellow, red, pink, whatever is there. the different colors of flowers and fruits is due to the presence of chromoplast, okay? And the third type of plastid is known as chlomo, the colorless plastid, which is called leuco. Leuco means colorless plast. This plast word is common, no? So leucoplast, chromoplast, chloroplast. So this plast is common in each and every word. So while learning, you can simply learn chloro, chromo, leuco. Chloro means green, chromo means colored, and leuco means the colorless plastid. Okay? Colorless plastid. Okay? Now next we have the leucoplast. Last one we will start. Leucoplast means they are colorless. Then what is the function where they are present? The function of leucoplast is to store the food materials in the form of starch granules, and they are present in different types of grains like the wheat grain, rice, maize. Where whatever food they store it in the form of leucoplast plastid. And according to the material which they store, three different types of leucoplasts are there. Like number one, if we say the word amyloplast, this leucoplast we are studying. What is amyloplast? It is the one which stores carbohydrate. The leucoplast which stores carbohydrate, that is called amyloplast. If it stores fats and oils, it is called elaioplast. The spelling is E-L-A-I-O. E-L-A-I-O. Elio, and the plast is there. Elaioplast. What do they store? Fats or oils. And the third one, if they store proteins inside them, the protein storing plastids, they are known as like amyloplast, elaioplast. The third one is also there called eleuron, like eleuron layer is there, eleuroplast. Eleuroplast. They are the one which store proteins inside. So protein storing leucoplasts are known as eleuroplast. Oil storing leucoplast or the plastids are known as elaioplast, and the carbohydrates storing are called amyloplast. Now next we have to start with chromoplast. Chromoplast are those plastids which give different colors to the different types of flowers and fruits. So let's start now the first one, that is chloroplast, and its structure along with the functions of chloroplast. Okay. Okay, now next one is chloroplast. What is chloroplast? The green colored plastid is known as chloroplast. And where it is found? It is found in all the green parts of the plant. And why it is green in color? It is green in color due to the presence of a pigment. Due to presence of a pigment called chlorophyll. The name of the pigment which gives green color to this plastid is chlorophyll pigment. Okay? Due to chlorophyll pigment, it is green in color. Got it now? Now, where is chloroplast found? I said it is found in all the green parts of the plant. Now, if you study the structure of this one, chloroplast, what is the structure of chloroplast? You will find that chloroplast is made up of double membrane, the outer membrane, this one, and then there is one more membrane, inner membrane. So I will draw the inner membrane also. Okay, so this one is called outer membrane. This one is known as inner membrane, outer membrane. This one is inner membrane. The gap between these two membranes, like this one, it is called periplastidial space. What is it called? Periplastidial space. Now, the inside part of the chloroplast, it contains a colorless ground substance which is known as matrix. So what is matrix? That matrix is called stroma. That matrix or the ground substance is colorless and it is called stroma. Then inside it, it contains many stacks. Stacks means piles of like this, thylakoids. Okay, so like this coils, thylakoids are present. they are arranged in a stack, in a pile, to form a structure called granum. G-R-A-N-U-M, granum. Like this, many granums are present within a cell. Okay, now see, here also I have drawn the granum. Now each of these granum are interconnected with the help of fret or lamella. What is it called? Fret or lamella. Now see, this is the structure, ultra structure of chloroplast. What is it? It is the ultra structure of chloroplast, in which you can see the different parts. Like first there is outer membrane, then inner membrane, then we have the ground substance which is called stroma, and inside the stroma we have the thylakoids like this. Each one is called thylakoids. Thylakoids, thylakoids. Just like the coins there. When they are arranged one on top of the other to form a pillar-like structure like this one, it is called granum. And this is the site of light reaction of photosynthesis, whereas stroma is the site or the place where dark reaction of photosynthesis occurs. Okay. Now the question is, how many chloroplasts are present in a cell? If you take one plant cell, the number of chloroplasts that may be present is, it depends, like it contains near about, you can say 20 to 40. Yeah, 20 to 40 per cell. So number of chloroplasts 20 to 40 per cell. Means one cell contains 20 to 40 chloroplasts. Okay. And what is it made up of? Where it is found? It is present in all the green parts of the plant. It is also found in euglena. You know what is euglena? It is a link between plants and animals, connecting link. Okay. And it contains different types of, like the size, it may be, the shape may be oval, it may be spiral, it may be ribbon shaped, it may be star shaped, depending on the organism in which this chloroplast is present. Like if you take a Spirogyra, it is a spiral shape. Okay. If you take some other organisms like euglena, oval shape. So if you take the different types of plants, different shapes of chloroplast is present. Okay. Now next, what is the number of grana within a chloroplast? The number of granum within a chloroplast, it is also mentioned to be near about, it is 40 to 60 grana. So how many grana are there? 40 to 60. In one chloroplast, the number of grana is 40 to 60. Okay. Now next is, what is the function of grana? It helps in the light reaction of photosynthesis. What is the function of stroma? It helps in the dark reaction of photosynthesis. So now next, what is the function of chloroplast? Number one, photosynthesis. You know that plants make their food in the leaves and green stems by a process called photosynthesis, in which 6 molecules of carbon dioxide combines with 12 molecules of water in the presence of sunlight, that is UV ultraviolet, and chlorophyll. It gives rise to C6H12O6, that is glucose, plus 6 molecules of oxygen is produced as a byproduct, plus 6 molecules of water is also formed. So this is the product, the glucose is produced. Now the second function, this is the equation, chemical equation of photosynthesis. The second function of chloroplast is it helps in phosphorylation. What is the definition of phosphorylation? The process of formation of ATP from ADP, an inorganic phosphate. I will write here ADP, that stands for adenosine diphosphate, plus IP, that is inorganic phosphate. I means inorganic, phosphate. 2 are already there, 1 more added in the presence of phosphate. The enzyme is there, the name of that enzyme is ATPase enzyme, adenosine triphosphatase, ASE. So in the presence of ATPase enzyme, ADP combines with inorganic phosphate to give rise to ATP, and this process is called phosphorylation. The third function is, chloroplast is a semi-autonomous organelle. Just like mitochondria is also semi-autonomous. Semi-autonomous means they contain their own DNA. Okay, so both chloroplast and mitochondria are 2 semi-autonomous organelles which contains their own DNA, RNA, and the ribosomes. Okay, so next we will study about the vacuoles. Okay, so next one is vacuoles. What are vacuoles? You may have seen generally in plant cells, very big empty spaces are present. Okay, these are called vacuoles. The empty space which are present in the plant cells are called vacuoles. Okay, large empty spaces are there. In animal cells, the vacuoles may be very small or almost absent. Like if I draw an animal cell here, the vacuoles may be like this, very small, in the form of food vacuoles. Like in case of amoeba, we have the contractile vacuole, which helps in the process of excretion, that is to remove the waste from the amoeba's body. So vacuoles, what is vacuoles? They are found generally in plant cells. Found in plant cells. In animal cells, they are absent or negligible. Negligible means very small in size. Now, the outermost membrane, the covering which is present around them, the vacuole, it is called tonoplast. So, if it is asked what is tonoplast, tonoplast is the covering of vacuoles or the cover of vacuoles. You can write the outermost cover of vacuoles is called tonoplast. Now, what is cell sap? The fluid which is present inside the vacuole that contains different types of food materials as well as the waste materials that are produced by the cell. They are stored here only. So that is called cell sap. So what is cell sap? The fluid present inside the vacuole. Fluid inside the vacuole is called cell sap. Okay. Now what is the function of vacuoles? Since they are present in the plant cell, they provide turgidity to the plant cells. They also help in osmoregulation. You know what is the meaning of osmoregulation? You heard this word? Regulation of salt and water balance in the body. What is osmoregulation? Regulation of, that is to maintain regulation of salt and water balance in the salt and water balance in the body. Like here it is the plant cells, so we can write in the plant cells is called osmoregulation. So function of vacuoles is it helps in osmoregulation also. It is the storehouse of different types of waste materials like tannins, resins, gums, alkaloids, okay, mucilages, and different types of salts like crystals of calcium, silica, calcium carbonate, calcium oxalate, and other secretory granules are also present. Now next is, what is cell inclusions? The next topic is cell inclusions. Till now you have studied about the cell organelles only. All the cell organelles we have finished, okay? The living parts of the cell. There are some non-living parts within a cell. Like plant cells, they do not have any special excretory system or the organs. Like we human beings have the excretory system, which consists of kidney, liver and all. But plants do not have any excretory system. So what do they do with their waste materials? Where do they keep? So those waste materials are the non-living things, which includes gums, resins. So non-living substance, you can write non-living parts of the cell is called cell inclusion. Alright, it includes gums, resins, tannins, latex. It also includes different types of inorganic crystals, like just now I said calcium oxalate. And now next we have the next topic, prokaryotic and eukaryotic cells. Okay, you have heard about the prokaryotic and eukaryotic. Their differences, at least 4 or 5 differences is needed so that you can write in the exam. What is the difference between plant cell, animal cell also? So first one we will do the differences between plant cell and animal cell. In your syllabus it is asked the differences, and only 4 topics are needed. What are they? Cell wall, centrosome, vacuoles and plastid. So first one is cell wall. Plant cell contains cell wall. So here you can write cell wall is present. Animal cells, is there any cell wall? No. So cell wall is absent. Cell wall is absent. And the second difference which we have to learn, we have the vacuoles also in the syllabus, centrosome. Only 4 references given. centrosome. Centrosome is the cell organelle which is found only in animal cell. So centrosome is absent in plant cell, we can write, and here we can write centrosome is present. Centrosome is present. It is found only in animal cells. Now third we have to do the vacuoles. So vacuoles, big and prominent vacuoles are present. So you can write here simply present, okay? And here you can write absent. Vacuoles are absent in, and if present also they are small or negligible, very small or negligible. And the fourth point which is asked is plastids. So what is plastid? You know, the one which gives color. So plastids are absent here. Plastids are absent in animal cell. Means only these four you need to learn, not the other differences. The cell wall, centrosome, vacuoles, and plastids. So here plastid is present. Plastids is present. In plant cell it is present, in animal cells they are absent. Okay, now last, what is totipotency and differences between prokaryotic and eukaryotic cells? So I will just say prokaryotic and eukaryotic. Prokaryotic and yet eukaryotic. Differences you have to learn, okay? Now what is prokaryotic cells? I will just say one by one all the points. Prokaryotic cell. Let's talk about the nucleus. Prokaryotic has a nucleus without nuclear membrane. It means the nuclear material is fully dispersed in the whole cytoplasm. Here they have a true nucleus with a nuclear membrane, and in prokaryotic cell the nucleoplasm is undifferentiated. Second point you can write, in prokaryotic cell the membrane bound cell organelles like mitochondria, ribosomes, lysosomes, they are absent, but here the membrane bound cell organelles are present. In prokaryotic cell the type of ribosomes which is found is 70S. Just now we talked now what type of ribosome is found, 70S, and here 80S type of ribosomes are present. Okay. Now prokaryotic cells are the one in which the cell division is mostly amitotic, but here the cell division is mitotic cell division which occurs. Then comes physiology. Respiration is carried out by the plasma membrane only, and here it is carried out by mitochondria, because you know that in eukaryotic, our cell is also eukaryotic, and respiration is done by mitochondria. Then by reproduction, prokaryotic cells like bacteria, blue-green algae, they reproduce vegetatively as well as by asexual methods. Sexual method may be present. Then eukaryotic cells can reproduce both by sexual as well as asexual method. So sexual process is present in eukaryotic cells. And the last one, the examples of prokaryotic cells, mycoplasma, which is the smallest living organism, that includes bacteria, mycoplasma, blue-green algae, that is called cyanobacteria, and eukaryotic cell examples are all plants and animal cells. Okay. Now one last topic and then this chapter will be over. What is totipotency? The word is totipotency. I just mentioned that word. Totipotency. Now how to define it? Totipotency means the inherent means the one which you get from the parents. The inherent capacity of an organism to regenerate the entire parts of that organism is called totipotency. The complete organism can be regenerated from some parts. In plant cells, you know that the meristematic tissue which is present at the growing parts of the plant has the capacity to regenerate the entire plant organism. Okay. Similarly, in animal cells, you know that it is the stem cell which is found in the embryo that has the capacity to regenerate the entire living organism. Okay. So up to here, now question answers, I'll give you the screenshot. You can try those answers and write them in your copy. That will be your homework. So that in the next class we can, you can prepare yourself for the test also of the first chapter. You write the question answers and be prepared for the online test. Thank you.